Everyone, thank you. So I will present our work on finding modules in a three-way time series data. So in this data, we have 3 dimensions. Rows are objects that we measure. This could be genes if we talk about gene expression data. This could also be brain regions in brain fMRI data. These are often called voxels or parcels. And we measure the activity of these objects over time in different subjects. And over the years, several methods were developed to analyze these data. For example, methods that co-cluster genes and subjects. Now, assume for now we have only one matrix, that is, we've measured only one subject. One interesting pattern you can search for are a subset of the genes and a set of time points such that these genes are highly activated in these time points. And of course, this reduces to the classic biclustering problem, and many methods were developed over the year. But the main question is, how do we find a consistent pattern across multiple subjects? So this is how the data looks like. So in terms of subjects, an interesting pattern will be one that is present in a large subset of the subject. So in our example, let's say we have subjects 1, 2, and 5, but of course we want to cover a large subset, but we do not require that it will be all subjects. In terms of the time points, now keep in mind we're talking about data of subject. This could be patients in the clinic, and we cannot assume that their biological response is synchronized. It means that we need to find the time points in each subject separately. But what about the genes? So of course, you can assume that the set of genes must be present in all subjects, but this will be very harsh. For example, think about a case where 2 genes are not active in a specific subject, or another case in which we have a complete pathway that gets turned on in addition in another subject. So this leads us to this example to account for this problems. Now focus in this example on the rows. The area A are the genes or the set of rows that light up jointly in the subjects. But we could have some missing rows. For example, in subject 5, we have most of the area of A, but some rows are missing. And the areas B, C, and D are some added subject-specific gene sets. So with this example in mind, we can now define a bit more formally what is a module in three-way data. So it has a core, which is a subset of the subject, subset of the rows, these are the area A, and we can have a different subset of the time points in each subject. Now the subject-specific part of the rows will cover some part of the area A, some part of the core rows, and we can have additional rows for each subject. Now how do we actually model the data? So in this work, we used a relatively simple two-group model. So we assume we have some F0 background distribution of the values in the data sets. And we're interested in modules that have extremely high or low values. So assume we now have another distribution F1 of the values within the modules. And for example, in binary data, it means that the probability of 1, we expect it to be much higher within the module than in the background outside the module. If you think about normal distribution, it could be that F1, for example, is a distribution with a much higher mean compared to the background. So in this work, we have developed a Bayesian generative model that explains the data, given the assumption that we have one module at a time. So this is the observed data. We call it Z. V is a measured object, T is a time point, and S is a subject. So I'm just going to give a brief intuition of how the model looks like. So we have a first layer of the module, determines what is shared across patients. Here we select the core module rows and the relevant subjects. In our example, we had subjects 1, 2, and 5, and we have the core module rows in the area A. Okay? Now, given the information that is shared across subjects, we can now ask in the second layer, what is subject-specific? So here we need to model the time points. So I'm not showing it here for simplicity, but we model time points using a Markov process where the basic intuition is that if we have a gene set that is highly activated and time point T minus 1 in a specific subject, then with some high probability, it will remain turned on at the next time point. Okay, so the second type of parameters will determine the actual rows in a subject-specific manner. Of course, which are the core rows that enter our module and the subject-specific added information, which again lead us to our 2 examples from before. Now, given this idea in mind, we now will define the posterior function that we try to optimize, and this will be the only technical slide in this talk. So we actually need to have parameters in the model. So in the first layer, we have two vectors, age and p. Age is just a binary vector that tells us which are the core module rows. And p is a vector of probabilities over subjects. So for each subject, we have a probability. And it tells us at which probability rows are actually being entered into the module of subject S. So just for intuition, if this score is 0, then this module is not relevant at all in subject S. The second layer will have two just binary matrices. C determines the actual time points that are selected in each subject, and edges determine the actual rows that are selected in each subject. Now, this is a Bayesian model, so all of these parameters have priors and hyperparameters, but I'm not showing this only to simplify things. So we can now write down how the posterior looks like. So we have all the parameters given the data, and it breaks down as follows. We have the probability of the hyperparameters that I omitted from this slide, but just I will comment that whenever we can, we use uninformative priors just to reduce the number of parameters. This is the first layer of the model, where we select the core rows and the subject relevancy. Given this information that is shared across subject, in the second layer, we determine the rows that are actually selected in each subject. And as I mentioned earlier, we model time point using a Markov process that is not present here as well. But given the HS and C, we know the rows and columns in each subject, which means that now we can write down the likelihood of the data, which is very simple. It's a two-group model. Cells within the module come from distribution F1, and otherwise they come from some background distribution F0. Okay, so we've developed an algorithm that we call twigs, three-layer module inference via Gibbs sampling. And we start with running some bi-clustering algorithm to get a good initial solution. And then we use Gibbs sampling to improve this initial solution. And of course, to use Gibbs sampler, you need to derive all conditional probabilities of each parameter given all other parameters. And this is a bit technical, and you can find the details in the paper. But the basic idea is that we have some initial solution that is not random. Okay, we ran another algorithm. And we use an iterative process of sampling that improves and gets us into a module with a high posterior probability. In practice, using simulations, we found out that roughly 50 iterations suffice to get into a good module, which is very, very fast. And as I mentioned earlier, we use uninformative priors. Up until this point, I just explained how we find only one module. Okay, so we would like to find more than one module in the data. So here we try two different heuristics. The first one is called filter. It's very simple. You run your bi-clustering algorithm, you get a set of bi-clusters, and you run the Gibbs sampler on each one of them. The second approach, we call it masker. It's also simple. You just run the algorithm at a time, get one module, run the Gibbs sampling on it to get a module. Once you have a module, you remove its signal from the data, and you repeat the process again. And the variation of these heuristics are commonly used in other bi-clustering algorithms. Okay, so in order to find out which heuristic works better and to test several bi-clustering algorithms for initial solvers, we ran some simulations. In this data, we have planted 5 core modules, each one with subject-specific information. And both in the binary and the normal data, we add a lot of noise to the data just to blur things and make it tough for the algorithm. And here we tested different algorithm. What is evident is this variant that uses the Bimax algorithm to find an initial solution, and the masker heuristic performs really well. So both in terms of the core rows that you detect, and the subject-specific rows and time points, where this is the Jakar score comparing the output of the algorithm to the known solution in the simulation. So from now on, we now have an algorithm that uses this variant. So now we have an algorithm we can finally apply to real data. Our first application is to gene expression data. We took gene expression data of patients after septic shock. So this is a very intense phenotype that can lead to death. In this data, they had 14 subjects that went after the septic shock, and for several days after the septic shock, whole blood gene expression data was measured. And we applied our algorithm in the binary case just to find out modules that have a lot of genes that are upregulated. So our algorithm detected 2 modules. The first one covers 11 subjects, and 53 core genes that were highly enriched for response to bacteria. The second module covers 7 subjects and 62 genes and was highly enriched to T-cell activity. Together, these modules cover all subjects in the data, so we get a nice fuzzy clustering of the patients into 2 different branches of the immune system. But this is only a part of what you can get from the output. Maybe another interesting thing that you can ask, what is subject-specific? So here we summarize the results in a network. We add a node for each patient. These are the nodes with the red stripes. The other nodes are pathways, and we add an edge between a pathway and a patient if this pathway is highly enriched in the module of this patient, but not in the core module. So it is not shared necessarily across patients. For example, here we can see response to fungus was highly enriched in subject 42 and subject 19. And if you just observe this network, you can get the idea that subject 92 and subject 24 are just major hubs. And a lot of these pathways are very related to specific responses in the immune system. So the insight now is that if we have a patient and we think that it is related to module 1, we can now ask whether or not it is starting to accumulate all of these pathways. Okay? So these 2 subjects are the only 2 subjects in this module that actually did not survive the septic shock. So if a patient that we monitor is more similar to this one, means that we maybe want to modify the treatment and take more care on them. So the second application was to brain fMRI data. Here we have resting state data of 20 subjects, and 464 brain regions were measured over 94 time points. And our algorithm detected between 4 and 5 modules, depends on pre-processing, but the results are very robust. What we found out here that the modules, the core modules, partition the brain into known brain regions that are related to resting states. And also you can see a nice spatial clustering in the brain in these maps. And we also get subject-specific enrichments that reveal some new connections between different brain regions. Just to give you one example, in this module we had 4 subjects that had additional enrichment for attention. Now this area is related to sensory stimuli. So the idea is that maybe these 4 subjects are much more attentive to the stimuli in the fMRI machine, which is quite noisy and not so comfortable. So it could be that these subjects are much more sensitive to the situation. Also in the brain fMRI, we found out that subjects cover a different percentage of the core module, so it ranges between 40% and 95%, and subjects have very different time points. So again, the times are not synchronized across subjects. So what have we learned by just comparing the brain fMRI application to the gene expression? In the gene expression case, we had very small yet highly enriched core modules, and subject-specific information could be substantial. We can have hundreds of additional genes. In the fMRI case, we had a different scenario where we had very large core modules, and each subject covers only a part of it, and the subject-specific information is relatively small, although we have some of it. In any case, in both situations, you really need this flexible definition of modules in order to find the complete picture of the data. Okay? We have also compared our algorithm to other methods that analyze 3-way data sets, although they find very different patterns, so we at least compared the methods in terms of the average coverage of the modules, and we found that our algorithm provides more than 2-fold improvement in subject coverage, both in the fMRI application and in the gene expression application. We have also, in the gene expression case, compared methods based on the enrichment analysis, and here we also compared to standard bi-clustering methods just to see how we perform. We rank all methods based on the number of enrichment that you get and the redundancy, and again, our algorithm was ranked number first. So to summarize, I've shown you a definition of what is a module in 3-way data, and we've developed a Gibbs sampling algorithm to find modules. Our algorithm outperforms extends method both in simulation and in real data. Coverage, a 2-fold improvement in subject coverage is a nice improvement. And we successfully applied the method to gene expression data and brain fMRI analysis, where we found that integrative analysis was crucial. And I also like to comment that there is lots of room for improvement, both in the model and in the algorithm. We can think about other nice additions to the model. So with this, I'd like to thank my advisor, Professor Ron Shamir, and Professor Daniel Icutieli from Tel Aviv University. I'd also like to thank the team that did the fMRI analysis with us from the Sourasky Medical Center, Professor Talma Handler, Nadim Aron-Katz. And I'm also grateful for Akamai for giving me a travel award to come here to ISMB. Thank you.
Questions?
How do you determine the number of modules that you extract at the end of the day? That seems like a model selection problem somehow.
Yeah. So we run, for example, in the masker algorithm that was selected, we run it iteratively until we get to a point that we don't find a module that is large enough or the F1 distribution is not interesting enough. So we define in advance a stopping criteria and we just use it later.
Thanks, very nice talk. Do you have any concept of maximality, for example, with respect to patients or with respect to other features?
What do you mean by maximality?
For example, if you include more patients in a cluster, probably you will have to lose some amount of the other features that you are by clustering. So, in a way, trying to maximize in one direction or the other.
So, I think it really depends on the patterns. Like in the fMRI data, we found out we now have new experiments on more subjects, and because this is rest, we do find the same regions over again. So if you just add patients, these are healthy individuals. So you do get very similar modules across many more subjects. In the gene expression data, we had a large heterogeneity of the patients, and then we can get this nice clustering of the patients into different biological responses. It would be interesting what happens if you add more and more subjects to the gene expression data, because it's fairly low number.
Great talk. I have a question about your masker algorithm. So you said you choose group core by first select and remove that. So is it a greedy approach, or is it can find a good enough solution?
It is a bit greedy in some sense that when we run the initial solver, we take its top score module. And then we apply the improvement and remove the signal from the data. So in some sense, it is greedy. Okay.
Hi, David. Thank you for the talk. You defined three-way data in this case with time series. Did you try to apply, for example, to subject times genes times tissue type, or
so
instead of using the time as a
no, we didn't.
dimension?
No, that's actually a very good suggestion. You can basically extend the model to have a different distribution in each tissue type and see what you find across tissues. That's
my fear would be that, for example, tissue expression could be so specific that you don't have that much of similarity that then you can get these high clusters. Or
so just in this, just to add, in this case, you really want to have different distributions across the tissues, but it's a very good suggestion. Thanks.
Okay, thank you.
All right, thanks again, David. And we move on to our next speaker, Benjamin Escott from Tufts, please.